Hey everyone, and thank you for checking out SPACEtomo. This is a little video explaining the different settings and the different functions within the target selection graphical user interface that is part of SPACEtomo 1.1. So if you're on a later version of SPACEtomo, some things might have changed. Maybe a lot of things have been changed, but this is the current state of SPACEtomo 1.1. So when you open your target selection menu, either from the command line, or it will be automatically opened by SPACEtomo if applicable, you will be greeted with this interface. You have an empty plot, you have a little column called target selection, and you have a little column here where you can load your map. And as you can see here in my maps folder, there's already 18 maps to be found. Those are all maps of yeast lamella. And you can either just select one from the drop-down and then click load map. You can click next map to load the next map in the list, or you have this little icon which has little thumbnails of all the different lamella. And as you can see here, it also already shows you what different state this lamella target selection process is in. Some of them have target selected and are already inspected. Some of them have no target selected but are inspected, and some of them don't have target selected and are not inspected. So the idea is that if your SPACEtomo main script is waiting for your inspection, that you go through all these lamella, look at the targets that either were automatically selected or you can manually select, and then mark them as inspected. So SPACEtomo knows, okay, these targets are good to go. Let's start the acquisition on those. All right, let's start with an example that you might experience when you run the automatic target selection with inspection, like this, for example, here. So this loading of the map might actually take a few seconds or half a minute, depending on your local environment or if you're accessing it via a network drive. But as you can see here, it loaded relatively fast, and we already have some selected targets here. You can go over these targets and you get little target information. For example, we have target 3 here that has a score, a SPACEtomo score of 0.68. and an image shift from the tracking series, which is marked in red here, of 11 microns. You can zoom in and out. This is quite responsive because it is all GPU based, and you get also a little tooltip if you hover over the plot that tells you exactly what you can do. You can drag targets to reposition them. You can shift and left click to add new targets. You can right click to delete targets, and you can middle click to add these orange geopoints, and those I'll go into a bit later. But for now, let's explore the rest of the interface a bit. So you can see we have some yeast cells here, and on the left we have all the classes that have been segmented by our yeast segmentation model. And for example, if we click on cell, it will take a few seconds, and then we load in the segmentation of the cytoplasm. And if we're interested in nucleus, we can click on the nucleus, and we see all the nuclei that have been segmented, the mitochondria as well. So this is how you can visualize the segmentations to inform your manual picking procedure. Alternatively, if you have already done the automated picking but you're not quite happy with the results, you can do a fresh target selection with all the same targeting parameters that you can set in the SPACEtomo script, but here you can actually play around and see the actual effect on an actual lamella. I also try to make a little tooltip, so when you hover over a setting, it should explain what it's supposed to do, and any more details about that are of course available in the SPACEtomo documentation. All right, so for example, let's set up a mitochondrium class selection. We want to avoid these classes here, and you simply add the names that you see on the left here, and you can separate them by a comma. All right, then we have a score threshold for sparse targets like mitochondria that don't fill the whole field of view. I would set this close to 0, maybe 0.1, around that area. If you have targets like the cytoplasm that can fill a whole field of view, that means that score can actually reach up to 1 if the whole field of view is covered, and then it makes sense to have higher threshold to avoid having tilt series collected on only maybe a third of the field of view covered in your target class. The penalty weight is how much you want to worry about ice chunks and the avoid classes. If you set this to 1, they'll be treated with the same weight as the actual target classes, which means that if you have equal area of target class and avoided class inside your field of view, the score goes back to 0. The maximum tilt angle setting will give you the exclusion zone where the target selection tries to avoid any kind of overlap. That doesn't necessarily have to be the same angle that you use for the data acquisition. If you have a lower angle, it will simply allow more overlap perpendicular to the tilt axis, which might be desirable if you're not too worried about having a little bit of extra exposure at the higher tilts. Then we have the image shift limit, and this is actually a serial EM property. The default value for serial EM is usually 15 microns, and most microscopes like thermoscientific Cryosys, but also Cryo M300s, are limited somewhere between 20 and 25 microns hardware-wise. So you could set this higher to about 20 microns without causing any issues, but better to try it out and see what happens first. For this, I'll actually set it a bit lower, maybe let's say to 5 microns, so I can show you what happens if there's targets that go beyond the image shift limit. We want to set up sparse targets for mitochondria. We don't want to target the edge, which means we want to have only half of the field of view covered. This might be useful if you target something like a nucleus where you are interested in a nuclear envelope and not in the nucleoplasm itself. And then we don't need an extra tracking target, which would set an additional target that is not necessarily on your target class just for the tracking purposes. All right, and if we hit select targets, we will select targets, and the details are not visible right now, but if you run it from the console, you'll actually get updated about all the little steps that are involved in the target selection, and it usually runs for several iterations. And here we are, it's already done. So we can see that it picked the targets on the mitochondria relatively reliably, and up here we have a good example where it shows that it tries to avoid all the black and ice areas. That's why this mitochondrium here is not completely centered. But let's say you don't care so much about it and you do want to center it, you can simply drag it and it will update the target accordingly. And once you change something, this save button here will become active. So these changes will not be saved until you actually hit the save button. All right, so let's center this one here as well because it seems to be a bit off. And actually, you know what? I changed my mind. I want my tracking area to be not on an actual target area. So let's set the tracking area over here and instead add a new target over here with shift left click. So this is how you can manipulate all your target areas within the target selection GUI. Next up, let's talk a bit about geopoints, these orange points here. As you can see, I tried to pick them automatically, but it only picked one in this example. And usually you want a couple more, at least 3, but preferably more like 4 or 5 if possible. And these will be used to measure the geometry of your lamella. SPACEtomo will move via image shift to all these geopoints and measure the autofocus, uses that as a z-value to fit a plane into the 3D space that should represent your lamella, and it will use this geometry of this plane to predict the specimen movement throughout the actual tilt series acquisition. So what I usually try to do is I set up some geopoints along the periphery of the lamella, so we have a large area that is covered that can be used to measure the geometry. And in theory, the more points you have, the more precise it should be, because if there's an outlier, it will have less influence on your actual measurement. Another thing that you might have picked up on is that we have actually 2 tracking areas at this point. And that happened because the extreme targets are further apart than the image shift limit you gave it, or that is the default in serial EM. And that means that when image shifting from one tracking target to the other one, it's actually not able to reach it within the image shift limit. And then it splits up the SPACEtomo acquisition areas into 2. So now we have one area with these 3 points here. You can see they're slightly lighter color, and if we hover over them, it actually says area 2. If we hover over one of the others, it says area 1, and they'll be all centered on this tracking target. I didn't notice this earlier, but it makes much more sense now to put this tracking target actually further down here, so these targets don't require as much of an image shift from the actual tracking area. All right, and once you are actually happy with your selection, you can hit save. And if you want to tell SPACEtomo that it can go ahead and set up these targets for SPACEtomo acquisition, you can mark it as inspected. And as you can see, this will lock all the changes. You can no longer select targets. You can no longer drag them around. It won't do anything anymore. You can also not add any more points or delete points. This is just locked down now. And then you can just simply hit next map, and it will load the next lamella map in your folder. So here you can see there's already targets selected and they're already inspected, so you cannot change them anymore. And now finally, let's move to a lamella where we have not selected any targets yet to see how it works to set up the target selection manually from scratch. This is an option now in the SPACEtomo 1.1 where you can completely skip the segmentation model, which is only working for yeast right now. And if you don't want to spend the time to train a segmentation model on your own organism, you can just run SPACEtomo with a manual target selection, which is, in my opinion, much more convenient than setting up the normal SPACEtomo right now. So let's say we move in here, we see, oh, this is a nice nucleus. Let's add a target here. Just shift and left click. And then we can set up a couple more targets here. And once we set up all the geo points, we can simply hit mark as inspected and we are done. Next lamella. So this is a much faster way to set up targets. The one thing I want to warn you about is that it's kind of important to have the record and view mag aligned properly. Because in this target selection mode, it will only box out a view mag image to align to and then start collecting the tilt series in record mode. So if the record and view mode are not aligned properly, you will be a little bit off target compared to what you picked here. This is a main difference compared to the SPACEtomo target selection, where you actually go into the record magnification to refine your target to make sure it will collect exactly where you intended to do. This is just something to keep in mind. But if your magnification is not extremely high and your calibrations of serial EM are done well, then this should still be very, very precise. Okay, that's it for the target selection GUI. This video will become part of a series of different tutorial videos for SPACEtomo. So if you haven't yet, check the YouTube channel for any other relevant videos. And if you run into any issues or have any suggestions for improvements, please feel free to start an issue on the SPACEtomo GitHub page. I would really appreciate that. Thank you so much for watching and good luck.